Here we go. Okay. So once again, Bioinformatics. We're talking about sequence data. So the programs there are a handful of programs that we will be using this week. Again, we will be using R because you can use R for like everything. And once again, like in previous weeks, honestly, you could probably, if you want to, use R for everything that we're doing in here. But we will use some other things that are also useful. So two other programs that I have up right now on D2L for you to go and grab. Eugene is the primary thing that we'll be using for a lot of things because it's kind of like this nice platform that integrates a lot of different programs into it, and you can use a lot of other programs from there. Bioinformatics kind of evolved in such a way in which there were lots of little programs that did one function, like one very simple function. Then people started like amassing them together, and then you have things like Eugene that has tons of functionality. It's all built on top of other programs that other people built, and then now they've essentially been pulled together in one interface, which is nice. Then we also have CView. CView does, there's nothing that CView in large view does that Eugene doesn't do, but it's more specialized and it's better at some of those functions, particularly with working with multiple sequences and sequence alignment and sequence editing. CView is a lot better at that than Eugene is. You can do all those things in Eugene. It's a lot more clunky and takes you like 3 times as long. So I like bouncing back and forth between CView and Eugene. So yeah, I can give both of those. And then there's this other package called Ape in R that we will be using somewhat as well. So I'd encourage you to go grab Ape. Go grab an Ape. Oh yeah, I didn't finish this up here. I should have finished my lecture yesterday. I thought, take a drink while I think of a definition here. So bioinformatics is essentially taking biological information and looking at it in the way that computer scientists would or data scientists would. And using principally sequence information. So principally we're using things like genetic sequence information or protein sequence information. But in some of the more broader definitions, we're also talking about other biological information such as metabolomics and transcriptomics and other things like this, where we have this information and then we're going to use it in some way to come to other conclusions or conclusions that are not necessarily directly apparent from that. We'll go with that for now. I might come up with a better definition for tomorrow. Wow, I got done early yesterday writing this. I was like, hey, I'm done early, I can go to bed. Oh, I should have looked over this once before. Okay, so really then this all starts with how in the world do we obtain DNA sequence information? And I'm going to start there and we're going to work up from there. So this will probably at the beginning overlap with things that you have talked about or done in some of the cell biology or else like there. And we're going to basically start with a little overlap there. So if a little bit of this is repeat. Actually, how much did you guys go over sequencing in cell biology? Any at all? Sanger sequencing? Okay. Is there any other class that you went over it in? Okay, so maybe, yeah.
I took a genomics class at WSU that was like all about that.
Okay, well cool,
Yeah.
hey.
Yeah.
So a little bit here and there. Okay, so maybe it's no overlap at all. So we'll start with sequencing. I'll keep in mind that there's basically no overlap with anything else, and I won't just blast through it as if you all know already. So, and really there are the 3 types of sequencing that are predominantly used right now, although there are tons of different. Sanger sequencing, which is kind of the old, venerable kind of sequencing. We have Illumina sequencing, which is also called chain termination sequencing, or fluorescent chain termination sequencing, or a couple other different things. Pyrosequencing, which includes a couple other different kinds that you'll often hear about. And actually, there's a ton more as well. So we're going to talk basically how we get these, because this is going to inform then how we view the data later and how we actually go through and understand the data later. And so we're kind of starting with this point and moving, kind of pushing past here. Yeah. And we'll start out talking about how does Sanger sequencing work. And this is the oldest method that we still currently use. It's been around for a very long time. And really, I remember learning about Sanger sequencing when I was a student here. I remember like, oh, well, I know they at least teach about Sanger sequencing. And I was reading through the others because most of these other ones have become prominent since I took cell biology. I was like, wow, all this new stuff is happening in the last 10 years. Anyways, so the key here, and the thing that is really interesting about this, is that it's using, instead of, normally when you do PCR, or you do something where you're replicating DNA, you add to it deoxynucleotide triphosphates, or DNTPs, and those are the building blocks of the DNA. Those are the base, essentially the base pairs that you're adding on. Well, Sanger sequencing adds in dideoxynucleotide triphosphates. And what these do is on that ribose, we have this ribose here like this. I wasn't going to draw this out. And then we have our base, and then we have up here, we have phosphate, phosphate. So phosphate, phosphate, phosphate. You guys did learn this in cell biology because I taught you this. So you guys did talk about this? Okay, cool. So, for those of you who haven't had it, normally you have in deoxy, you have a proton here, and then you have a hydroxyl group there. So this is the deoxy right here. Well, if it's dideoxy, we also have, instead of a hydroxyl group there, we just have a hydrogen there, which means then, if this gets incorporated into your chain or into your stranded DNA, it can't extend beyond this point. It won't go any further at all. And so we add in these dideoxynucleotide triphosphates, and we do this at about 1% of all of the rest of them. So actually, most of the time, you will grab a normal nucleotide and stick it in there and keep going, but every so often, you're going to hit one of these chain terminating ones and stop extension of your stranded DNA at that point. These ones that are dideoxy are also fluorescently tagged, at least in modern Sanger sequencing. What they actually used to do is have different reactions, one where you had the adenosine dideoxy, one that had the cytosine dideoxy, one that you had the thymine dideoxy, and one with the guanine dideoxy. And then talk a little bit about how we did that. But in modern times, we do them all together, and they're each fluorescently tagged, with generally a different fluorescent color for each different base. And then we do something like PCR, where we're extending the, I'm not going to go over PCR because hopefully you talked a lot about PCR in cell biology. Yeah. And so essentially we have our template, our primer, and we're extending. Now we're extending. At some point, one of these is going to be incorporated in, and it terminates extension. We stop there, but it's also tagged fluorescently with whatever is incorporated there. So, for instance, if we have a green fluorescence, we know perhaps it's guanine or something else like that. And then all of this is run out on a gel to separate these amplicons by size. So everything, so for instance, imagine here now. Imagine now that we get one base, two bases, and then we get the third base is dideoxy, so now we have like a red fluorescent marker there. We have another one that goes a long ways, whatever that is, and then we get like a yellow fluorescence there, so we know that is like something. Another one that goes out like immediate ways, and then as guanine there. Another one, blue, whatever that is. And then we run these out on gel electrophoresis so that they all are separated by size. So we have the smallest, the next smallest, all the way up. So essentially we will have, because there's enough of these both the nucleotide triphosphates and the dideoxynucleotide triphosphates, and we do this enough, we should have a terminator at every single possible position in there. So we have some of them that extended 1 stop, some that extended 2 and stop, all the way down out to however long our entire chain is. And so we will get these all separated out, and then we can fluoresce this, and we will see them light up according to the color of the fluorescence that is tagged to the particular dideoxynucleotide triphosphate. And then we can just look at this and then read out the sequence down the gel. Now, in olden days, before we did this, we actually would run out, there would be, the only tag would be like, for instance, a radioactive tag, and we'd actually run them each out on a different lane. So we'd have an A lane, and a G lane, and a T lane, and a C lane. We would have band, 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 band. Like this, and then you'd have to like look down the gel and say, okay, so A, now it's a G. We don't do that anymore because, hey, we have fluorescent tags now. They didn't have that back in the 60s and 70s when we started this. And so we've done it this way. And also, instead of running them out on complete gel, they're running them out on capillaries. So you have a long, thin tube that is filled with the gel. And we're running actually a fairly small amount down this capillary. And then we just have something that runs down that and reads that. So at that point, we have just a sensor that runs down the capillary and reads the fluorescence at each position as you run down the capillary. So essentially, to give you a more visual termination of this, again, we have our dideoxynucleotide triphosphates. They've each been tagged with some sort of fluorescence. And then we extend and we have those incorporated at all these other different parts. And then they run them out on a capillary gel, and then we have some sort of detector there that is going to go through, detect each of the bands that we have, and that comes out in this thing called a chromatograph that looks like this. So this is actually the output that you often get from sequencing data from Sanger sequencing. So if you go along here, what you'll see is you have the different colored bands, and then we can call the bases by where we're reading all these. And this is, if you have a good sequencing rung, this is what it should look like. If you don't, all this background starts getting kind of high and crazy in there, and you will see examples of bad sequencing before this class is over, trust me. So that's essentially the output that you get from this, and then you read the sequence from that. Are there any questions on that so far? So Sanger sequencing has some advantages still to this day. The first thing is that it's good for small projects. This actually works a lot better for smaller projects, like for instance what we're doing in this class, than a lot of the high throughput methods. It also allows for very long individual reads. So every time we're actually reading out through one read of DNA, or essentially reading one, not necessarily one DNA molecule, but all the products from one DNA molecule or a few DNA molecules, we can read them out to a long distance. So actually the read length is between 400 to 900 base pairs. That's what you can usually get, and actually, to tell you the truth, you can get farther than that now. Actually, I think we can get reliably, pretty reliably, up to about 1,500 to 2 kilobases. So it's actually better than this now. But generally, you get one read per run. And in modern Sanger sequencing, we can parallelize this a little bit, so we can get like tens of reads per run, but it's not a ton. However, the accuracy is very, very high. If you have done all the steps leading up to actual sequencing well, if you had good amplification or something else like that, the actual accuracy of Sanger sequencing itself is very high. It's like 99.9% accuracy. It's very good. And for a single run, it actually doesn't take that long, less than 3 hours. So that isn't too awfully bad for an individual run. Now, all that being said, you're only getting very few reads per run, so that we don't have a parallelization here that helps out, but we got that. And the cost for 1 megabase, so 1 million base reads, is about $2,400, which is really insanely expensive for one set of megabases. Now, to give you an idea about how much this is actually how we did the original human genome, and it makes sense with this technology why it costs so much. Even today, if we were to use this technology to, from scratch, try to sequence something the size of the human genome, I think they announced when they had 3x coverage, which is actually still pretty crappy, which means that at each individual base they had it sequenced 3 times. It would still cost you somewhere in the neighborhood of $23 million to sequence the human genome. Now, it costed them way more than that, but then again, they're using 90s technology. But even with technology today, it would still cost insanely much to sequence a genome the size of the human genome from scratch. Now, all that being said, when we actually sequence your genome for $1,000 now, we're using different sequencing technology, number one. Number two, we also have a sequence in place that allows us to do shortcuts that we wouldn't do otherwise. So it's not exactly the same doing it from scratch as doing it when you have a genome in place. Yeah.
When you are doing it from scratch, how do you design the very first primer you're going to use?
Okay, so actually you don't. You use adapters that you ligate onto the beginning, and then you prime to those adapters. So you're actually not using primers. Yeah.
So what are the adapters?
The adapters are something that is some set sequence. It honestly, at that point, I think it's not really important what the adapter is. I could be wrong about that. But yeah, it's just a short sequence that then you can ligate onto a blunt end. And then you know what the sequence is there, and then you prime to that, and then you can start extending there.
So why is there the extra step instead of just designing a primer that will?
Because you don't know the sequence.
But don't you mostly because of telomeres?
For the entire chromosome, but you can't read an entire chromosome.
Oh yeah.
Yeah, we have to read short little chunks at a time. Yeah. Anything else? All good questions. Okay, let's move on to another one. So there's also Illumina sequencing, which is also known as reversible dyed terminator sequencing, sometimes as well. And then this is usually done for a lot larger sequencing projects with a lot larger parts of DNA. I'm going to fly through these kind of. And then the DNA is prepared. You can either do this sometimes by PCR amping, but also if you have a much longer set of DNA, it can be done by what's called tagmentation. We use transposomes to cut up the DNA into smaller pieces. And again, in this case as well, you have an adapter attached to both ends of this DNA. So now we have a known sequence that has been tagged onto both ends of this DNA. And then we hybridize it to a flow cell long in a lane. So let me illustrate what this looks like. So now we have, in most cases, a set of DNA that has been chopped up in some way. So we have it in little short chunks. And now we have a flow cell that looks a little bit like a glass microscope slide. In here, we have all of these lanes, which is a lot more than this, that then DNA is flowed across here, and then you have these short little oligonucleotide long that is sitting kind of down on here that is then attached to the flow cell. As it goes through now, the adapters that we have ligated onto these chunks of DNA attach to these oligonucleotides that are down onto there. And then, so now at each point, and they've done a dilution, you've done a dilution, in which case you're not actually filling this up. Very few of these are actually going to be attached to a piece of DNA. So you have one here, maybe one here, one here. So very few of them are actually attached. And then using something similar to PCR, essentially PCR, you are going to essentially make a colony of these by PCR. Now, I'm going to show you an illustration that goes into that a little bit more in depth, but just hold on for a second. And then we're going to use dinucleotide triphosphates with reversible inhibitors and fluorescent tags to amplify this one base at a time. So, if you're looking down one of these flow lanes like here, looking down it like this is essentially what you would see. It's these small and two different kinds of oligonucleotides going down here. And one of these are going to be complementary to the adapter. Actually, both of them are complementary to the adapters, but these are complementary to the adapters that we have already put onto these chunks of DNA, and they stick to these. And once you're at that point, now you have this chunk of DNA that has the two different adapters on it, that's complementary to this, and it sticks there. But it's not just going to stay there. So again, remember, we have like a few of these per every one of them. It's going to have its other adapter hybridized to one of the other oligonucleotides down there. So now we have this folded DNA like this. We then go through essentially PCR, where we have this serves as our primer, and we have a polymerase, and then we extend it. And now we have two, and both of them are also attached to this flow cell. So now we have two of them. And then we go on and we keep doing this till we have somewhere in the neighborhood of about 1,000 copies that are colonies or exact complementary replicas of the original piece that got stranded there originally. So now we have, if we look at this flow cell, little points that are colonies that are replicas of that piece of DNA. And once we get to this point, actually, what we do is there is other reagents that we add that are going to clip half of these. So we actually only have one direction left. So we're going to clip off the rest of those. We only have one direction left. And then from there, we're going to go ahead and start extending one base at a time. So again, these have reversible inhibitors on them. We attach one nucleotide, it stops the reaction there. We get fluorescence. After we get the fluorescence, so we can read, again, like we do with Sanger sequencing, we can read what base was added. We remove the inhibitor. We attach the second nucleotide, again, inhibit, so it extends one. We read that again. And in that way, we can determine what it is, or what the sequence is, for each chunk of DNA that's attached to every point along the flow cell. Now the advantages of this is it's very high throughput. And actually, I was just looking up what the most recent things can do, and you can get up to about 6 billion clusters per flow cell, which is tons. And you only need one copy of each target that you're running. You don't need to have this amplified beforehand so that you have amplified every single last, like in Sanger sequencing, you need to be able to amplify every single last transcript that you're going after. You don't, in this case, before you run the sequencing. And this makes it very automatable, as it is with most. Now, the problems with this, or maybe not problems, but one of the realities of this, that you get very short read length compared to Sanger sequencing. Only 50 to 300 base pairs per each read can you actually do. So when you go through your process of tagmentation, you are chopping it up, hopefully, into smaller chunks than that. Or about that. But again, you have a lot of reads per run, like up to 6 billion reads per run, which is a lot. And again, its accuracy here is really high. So for every single run or read that you're getting, you're having really high accuracy. However, the runtime can be quite a while. You can run up to, actually I think this is better now looking around, but at least over the last few years, it could be up to like 11 days per run. Well, that being said, you're getting 6 billion reads in 11 days, so that's a lot better than you can get with Sanger sequencing. Now the cost of this compared to, compare this, yes?
What's the purpose of having lots of reads per run?
The purpose of having lots of reads per run then is getting just more sequence data in a shorter amount of time. So we're only getting 50 to 300 base pairs per read, so for every strand of DNA that we're sequencing, but we're getting 6 billion of them, so we can sequence more of a genome or something else like that in a much quicker amount of time. So we're doing that in like 11 days rather than Sanger sequencing that'd take forever to do that.
So when you do this, you just like digest DNA you're interested beforehand, so you have a bunch of
Yeah, so that's what we were talking about back here with segmentation. Okay, I'll go back to. We're gonna cut it by transposomes, and that, yeah, we chop it up before we do this. So you take your genomic or mitochondrial DNA or whatever you've isolated, and then you chop it.
Do you have to like be careful to chop it into chunks that are about the right length, or do you just kind of?
Well, if you chop it up into too small chunks, you're not going to get reads the size that you want. But they can be. Well, one thing here is you're probably going to do several. If you're going to actually sequence the entire genome, you're not going to run it once through Illumina sequencing. You're going to do it several times, probably more than several times. And so actually, they can be longer. Is fine, because then you'll still turn it. You won't get the entire sequence read out, but that's, as far as I know, that's okay. Like having longer.
Okay.
Yeah. Right. Okay, and then the cost is way cheaper for how much you're getting. So for 1 megabase here, we're talking like 15 cents or less compared to 2,400 for single sequencing. So like the cost difference between this and what was happening before is really, really crazy.
So each circle that you drew up there, that represents a different slice of DNA that has been replicated.
Yes.
over time?
Yeah. So a colony of DNA that all descended from one original
Okay.
chunk that we chopped out.
And then basically you're just doing the same principle as Sanger sequencing, just on a bunch of different.
Kind of, but in Sanger sequencing, the dideoxynucleotides, and this is one of the big differences, is that's not a reversible termination. In this case, the termination is reversible. So like it'll terminate, but then we can reverse the terminator so it can continue to extend after that. Does that make sense?
So that's why you only need one copy of the target, or.
You only need one copy of the target because you're amplifying on the flow cell.
Oh, okay.
Yeah.
Okay. So you still need to amplify it in some.
Yeah, but it's being amplified actually. I'm talking about you don't need to amplify it before you start your actual run. So this is really nice. Like for instance, when you see people doing, like for instance, they go and they look for environmental DNA, and they're looking for just single copies of a particular kind of DNA on, in like the water or something else like this. They're gonna use something like this rather than like PCR and Sanger sequencing. Yeah. Yes and yes.
So how does like how do you reverse the termination of
You know what? I'm not totally sure.
okay.
I didn't read that deeply into it.
So,
I know the basic principle, but I don't know enough of the nuts and bolts to know exactly how that works.
I expected you to build one of these things, Kurt.
I'm sorry.
So does it add like just one base pair and then read the whole thing?
No, it just adds one base pair, and then we have the fluorescence of that base pair, of that base that was added.
Okay, so like the machine that's doing this like allows one base pair to be added to all the different colonies, and then.
Well, it's not that it allows it because once it adds, it can't add anymore because there's the terminator that will get removed.
Okay. Are all of the nucleotides in there to be added? Are they all terminators at this point?
I believe so.
Okay.
Yeah. And Kalia and then right.
How do you put them in order? So do you have a bunch of really small pieces? You said like we could do the full like genome, but like how would you know the order?
That is a very good question, and we will get into that. Essentially, what you have to do is you have to use other technologies and essentially line them all up. And essentially what you're doing is you're not doing this once, you're doing this multiple times. And so this is the process of genome assembly. Then you have this, and then we have hopefully another one that overlaps, and another one that overlaps here, and the one that overlaps here, and overlaps here. So you have all these overlaps then that you can then start to put the puzzle together. And this is why you can't assemble with just one single read through an entire genome. You at least have to, at the bare minimum, you have to have, if everything works exactly right, 2x coverage to actually assemble if you don't have a reference genome to go off of. So this is one of the things that makes it simpler when we go for your genome, is that we don't need the multiple coverage to assemble your genome, because once we get a transcript, we know basically where it goes. Yes, Ryan.
What is a flow cell? And how is this data read? That's what I'm
Okay, two really good questions. Okay, so a flow cell is essentially just, I believe it's glass or plastic that you have these lanes in, which are essentially grooves, in which you then have all of these, all of the nucleotides that have been bound to that lane, that are then complementary to the adapter that you've put onto the DNA. And then how it's read is that you just have something like a CMOS or a CCD chip below here that then reads the fluorescence as they fluoresce. So, actually I don't have a picture of that. So, as we add one, so imagine to this colony here, we add one, right? And then we excite it and it fluoresces, and we have a certain color. We have below this now, just some sort of optical sensor like you would have, like a CCD, that you would have in your camera or something, that then reads, of course, I think they're much, much higher, not density, but they're a lot larger. But then they're reading then the individual fluorescences that you get here from these.
And they're unique for nucleotide?
Yeah, so each fluorescence, fluorescence that you're going to get is different for each nucleotide. So then you tell the nucleotide by what color it's fluorescing. Yeah. Any other questions?
So this reads like 300 base pairs over 11 days. So it
Times 6 billion.
Times 6 billion. Yeah, but then
Up to
these are
6 billion.
6 billion like in parallel.
Yes.
So it's got to take like a few hours per nucleotide or something.
I'm not exactly sure. I don't know how much of that is the process before that.
When people say that they have done high throughput sequencing, is this what they've done?
One of them, yes. There's actually several different kinds of high throughput sequencing. There's lots of different kinds of high throughput sequencing. This is one of the more common ones. The other one is Pyrosequencing. Thank you for the good segue. Which one version of this is also, for instance, you'll hear about 454 sequencing, which is also a type of pyrosequencing. Yes. So this operates even a little bit differently. The last two that we talked about dealt with fluorescence, and this one is not going to be dealing with fluorescence. It's actually dealing with light production. So we don't have to excite it. It's actually light it being produced on not quite its own. But it's going to essentially come from this system right here. Is that okay? So we have nucleotide triphosphate is going to get added. We only keep one of these phosphates, and we have this pyrophosphate that is going to be released from the reaction. Hence, and we're going to be using this pyrophosphate now that is released from this reaction to do this, or to actually power our sequencing, so to speak, and hence why it's called pyrosequencing. So we add a couple things. One of the things that we have added to this as we do this is ATP sulfurylase, which then takes adenosine phosphosulfate plus your pyrophosphate and is going to make ATP from that. Then we take that ATP plus luciferin plus luciferase will make light. Okay, so the point here now is just that we can now connect, or we can link the addition of a nucleotide to the production of light. And that's essentially how we are going to get from adding a nucleotide to somehow detecting the addition of that nucleotide. Okay, so backing up now, how do we go about this? Again, we're going to have an adapter added to the template or the chunks of DNA, and it's going to go through again some sort of generally digestion like we've done before. We're getting into smaller chunks. We're going to add an adapter, just ligate a chunk of DNA of known sequence onto there. And then that template then is going to be hybridized to a bead that has oligonucleotide with that. So, and in doing this, I'm not exactly sure the process of it, but essentially each bead is going to have a different sequence to be sequenced. And then we extend one base at a time by only adding one type of nucleotide at a time. So again, we will add essentially just the adenosine triphosphate or the guanine triphosphate, and then we add the subsequent things as well, and we'll get light created. And then we use a pyrase to remove the DNTPs so that the entire thing is clean again. We do not have the nucleotides remaining there, and then we add the next possible nucleotide. So for every position, we have to go through this whole cycle of adding each set of nucleotides. So, essentially we have our DNA. We're going to chop it up, denature it, and then chop it up. And then we add the adapters to that. Each of the adapters then will bind it to a bead, and then we get those beads into different cells that we're going to be looking at. And then as we add our different deoxynucleotides, we're going to come in here and they will be added sequentially to all this. This will release the pyrophosphate, which then goes through our system that we have that produces light, and then that light is going to be able to be detected. Now, you can't see this quite here. This is essentially the output from here. One thing that you'll notice, however, is if you have a string of two of the same, those will both get added. We don't have a terminator here that's going to stop it after one addition. So what you will get then at that read then, this is hard to see here, but you'll have single peak, a single peak, a single peak, adding. But if you have a double peak, that means that we have added 2. If we have a triple peak, then we have added 3. Because again, as we add more, it's going to, essentially, as we're adding twice as many, we're going to get twice as much pyrophosphate, which is going to lead to twice as much luciferase reacting with the luciferin and the ATP to produce light, so you get more light produced. And so that is how, in essence, this is working. And we do this iteratively with additions of each nucleotide triphosphate, or deoxynucleotide triphosphate. Now, again, this is nice because this can be high throughput. Yes?
Real quick question on that previous picture.
Yeah.
Does it ever hit like an extent where it cannot quantify? So like if you had a string of 10 or 12 A's in like a poly A tail, would the program be unable to distinguish that many, or would the peak just like dwarf the other peaks?
It would dwarf the other peaks. It would be really big. I don't know if it gets to that point where it can't quantify anymore. My hunch is no, because we tend to be pretty good at quantifying amounts of light. And so if it's, and this is quantized, so it's going to go, it shouldn't get, hypothetically, it shouldn't get fuzzy as you add more, because you are adding it in multiples of what one is. So my guess is that you should still be able to quantify it, but I don't know exactly for sure.
How does it track the DNA polymerase? How does it move it along?
So,
So it's not.
well, it stays on there, so it's ready to add another nucleotide but it can't go until, like, for instance here, it can't add it until it has deoxyadenosine triphosphate. It can't add anything. So it's going to wait until it has that available, and then it will add. And we know when we've put that in, and so if it lights up when we've added that, then we know, hey, there's a T there at this next position. And actually, you go 2 for it, and it'll be a double peak, and so we know, oh, we added 2 of those, so we know we have a TT at that position. That make sense? No. Yeah.
So does it have to like thoroughly rinse the beads each time so that you don't have like leftover nucleotides from previous iterations?
That's what the pyrase does. So we're going to add an enzyme that breaks those down. And enzymes do things better than washing. But this is also, I think there are washes that happen in here because you can't just add things infinitely. And that's one of the big reasons these are on beads and not in liquid media, so that you can wash without losing stuff. Any other questions? Okay, so the advantages of this, number one, it's high throughput. Like we see before, we can parallelize this a lot. And the read length is actually relatively large. We can get up to about 700 base pairs per read. And we're not incredibly high like we were with Illumina. We can do this up to about 1 million reads per run. So we can do this, parallelize this up, anyways, up to about 1 million reads per run. So we can actually sequence about 700 million base pairs per run. And again, we have really high accuracy with this, like nearly 100%. And the runtime is a lot better than Illumina. We're not doing it as many, but our runtime is less, about 1 day per run. And this is going to cost about $10 per megabase. So it's intermediate. Essentially, it's intermediate in both of the things. Now, this was one of the first generations of high throughput sequencing. So it doesn't perform nearly as well as like the Illumina or some other things like that, but it still does pretty well. Another thing that's kind of annoying with this, from what I've been told by other people who do it, is that you're also taking 4 enzymes. Like, rather than just using DNA polymerase, which we use in Illumina, like one single enzyme in there, it's taking, we have the polymerase, we have the sulfurase, we have the pyrase, we have the luciferase. Am I missing any? No. And so you're dealing with a more complex system as well, which makes it more prone to not work or have bugs. Anyways, but there are more. So Sanger, Illumina, and Pyrosequencing are all sequencing by synthesis processes. We're adding on nucleotides and detecting the addition of a new nucleotide, and we're doing it that way. However, that being said, there's also sequencing by ligation procedures. One that is being used somewhat now is called solid sequencing. Wow, that went by very quickly. I'll finish up with this just real fast. We also have one that's called single molecule real-time sequencing. I actually don't know anything about this other than it exists and a few things about it. Number one, it has really high read length. You can read up to 4,000 base pairs in a single read, which is crazy. And you can do something in the neighborhood of only, but the other hand, is that you only get about 500,000 reads per run. Now the nice thing about this is going to cut down your post-processing time. The smaller that you chop up individual sequences, the more processor time it's going to take to reassemble those. And the more reads, the more coverage that you have to get to actually get those to all chunk together. If you get higher reads, you don't need as much coverage, and it's going to take a lot less processor time to actually reassemble a genome or something like this. The other problem is that it has low accuracy, so it's only about 87% for a single read, which is annoying. I guess I've never actually used it, so I don't know if it's annoying. But again, you would have to. This does increase your need for coverage because you have the low accuracy. Okay, so the final result of this is that you're going to, from this sequencing, you're going to get, whatever you use, a string of A's, C's, T's, and G's. You're going to get something out like that. And this can be read out a couple different ways. In Sanger sequencing, you can get a raw chromatogram, which is a .av1 file, or the sequences themselves are generally stored as a text file in one of these 4 formats that are the most common: FASTA, FASTQ, Nexus, and Filet. Anyhow, with that, Wednesday, not tomorrow, we're going to start going into the actual sequences you get out themselves and how do we work with those and what do we do with them. So, okay, with that, enjoy your Monday.